Good evening. My name is Teresa Müller. I'm from the Bioinformatics group of the Computer Science Department of the University of Freiburg. Today I would like to talk about the locality dilemma of Sankoff-like RNA alignments. Sankoff is the golden standard for simultaneously sequence and structure alignment. If you have two input sequences, for example two non-coding RNAs, the Sankoff alignment will simultaneously compute sequence and structure via a dynamic programming approach. And the result will then be the optimal sequence structure alignment. Due to its high time and space complexity, Sankoff is not very feasible for expensive computational tasks. Therefore, there exists many heuristics like Locana. Locana computes the base pair probability for the structure alignment where the Sankoff alignment originally uses the minimum free energy. Locana can reduce time and space complexity and is therefore more feasible for computationally expensive alignments. It also can perform local alignment, where you have a local motive embedded in some context. And the output then will of course also be a local alignment where just parts of the input sequences are aligned. But you already see here that there might be some problems. And that's what I want to investigate with you now a bit further. So what could go wrong in a local Sankoff-like alignment? Performing a local alignment, you always have a local motive which is surrounded by context. The local motive you see here in blue, and the context is depicted in orange to have a clear distinction between both. You see that the local motive and the context both contain possible structures. And the structures in the context can also be of high similarity. Therefore, the predicted alignment could be extended into the context so that the local motive is elongated. Another difficulty could be that some parts within the local motive are less conserved structure-wise. Therefore, the local motive could be too little. The third possibility could be that inside the local motive there are structural parts that seem to be more conserved, and this leads to structural misalignment. So as we saw already for global Sankoff-like alignment, the alignment quality overall is quite good. We might have some misaligned parts, but overall we get quite accurate results. This is not the case for local alignment. And therefore, we want to investigate now or have a look into the structure or the structure component of the score. To do this, we choose Locana specifically because Locana has a clear distinction between the structure and the sequence contribution inside its objective function. For example, if you have an alignment like here, the Locana would score the structure contribution, so parts of the alignment where structure is aligned, which is depicted in our example with a gray background. You see that the base pair score is weighted by the structure weight, and the ribosome score, which scores match and mismatches, is scored by the tau factor. And parts where you only have sequence alignment inside your sequence structure alignment, these parts will be scored by the sequence contribution. So here again you have your ribosome score and the gap opening and extension penalties, which are depicted in the blue circles. What you should remember from this slide is the structure weight, because the structure weight controls how much of the base pair scoring will be influencing the objective function and therefore the final result of our alignment. So now we dive into our results. First, we computed the normalized Locana score for increasing structure weights. And you see this for non-coding RNAs in green, for shuffled non-coding RNAs in blue, both calculated with the sequence structure score, and for a sequence-only score, which is depicted in the dashed line. The score is normalized by the sum of the sequence lengths, so we have a normalized score per position here. And when you think about local alignment, you always have to remember that you need a negative expected score of random sequences to know the boundaries of your alignment detection. So whether you should align this part because the score is still increased, or you have found the border of your alignment prediction, and the next parts would be then the context, which would be aligned. And we know this negative expected score holds for sequence-only alignments, which you can also see here by the dashed line, but it was never thoroughly investigated for the structure contribution. So you see here between the green line, so the non-coding RNAs, and the blue line with the blue boxes, that Locana can clearly distinguish between non-coding RNAs and random sequences. And this even holds for a structure weight of 200, which is Locana's default. However, if you compare high structure weight, the shuffled non-coding RNAs of the sequence structure score and this sequence only alignment, you see that the sequence structure alignment gets positive, which of course we don't want, and shows this positive contribution when you increase the structure weight. So we see that we have a positive bias within the score due to a positive contribution of the structure weight. Next, we wanted to investigate the local alignment length growth, because this positive structure contribution would need to have some kind of influence on the local alignment. For this, we have generated a set of random sequences of length 100, and you can see here for an increasing structure weight, the expected alignment length, again for the sequence structure alignment and sequence only score. For a structure weight of 250, you see that already more than half of the sequence is aligned, and comparing this to the sequence only score, where less than 10% or roughly around 10% is aligned, shows you that we have an increase of the alignment length of random sequences when you increase the structure weight. And this increase of alignment length could also hinder a correct boundary detection. Therefore, we next try to have a look at the boundary detection. And we did this by investigating the sensitivity and specificity. When you look at the sensitivity, you see how much part of the motive is actually detected by the alignment, and the specificity will tell you how much of the context is not predicted within your local alignment. We did this analysis for sequences with a low sequence identity, so less than 70%. You can see that for an increasing structure weight, you will also find more and more of the local motive. And you can also see by this figure that you need, especially for sequences with low sequence identity, a high or a higher structure weight to at least predict half of the motive. If you go, for example, to structure weight 50 or 100, in most cases you find less than half of the local motive, which in the end you also don't want to achieve when you do a local alignment prediction. Looking at the context, so the specificity on the right side, you see that by increasing the structure weight, more and more of the context will be also aligned, so your alignment is extended into the context. So to summarize again, we saw that we have a positive score contribution due to structure. This leads to alignment elongation, and we have a trade-off for setting the correct structure weight between finding the complete motive and an extension into the context. But of course, we did not want to stop here. We did not want to singly talk about problems. We also wanted to find a solution and improve the prediction. Therefore, we did parameter optimization first of global alignment. Here, although the global alignment was working quite good already, we found a parameter set that improved on our data set the alignment quality. But taking this alignment, optimized parameters and using it for the local alignment prediction did not reveal convincing results. Therefore, we did the same parameter optimization, but this time for the local alignment. And we could really see an improve in the alignment detection, much bigger than the one of the global alignment, because the quality overall was lower than for the global alignment. Comparing now the final optimized parameters also showed us that what we have seen before was also reflected in the optimized parameters. Because, for example, if you look at the fourth column, the structure weight, for the global optimized parameters, we have 210, which is around 200, the default value. But for the local optimized value, it's 115, which is convincingly lower, which could result out of this problem that having a high structure weight leads to an extension into the context, which lowers the quality of the local alignment. And also the gap opening and gap extension penalty is increased. So we have an increase of the penalty and a decrease of the structure weight. But we have already seen that especially for sequences with a low sequence identity, one would like to have a higher structure weight to detect the complete non-coding RNA or the complete local motif you want to find. And we thought about a solution for having the same set of parameters for global and local alignment. And we propose a position-wise penalty for this. If you look at the objective function, you see that the penalty is really per position. So you take -4 times the penalty for the structure contribution, because of course you have here a base pair, and so you align for position. For the ribosome score, where you have match or mismatches, you have to reduce 2 times the penalty, and for a position aligned to a gap, you have to reduce 1 time of the penalty. And when we look now again to the local optimized parameters, but this time with position-wise penalty 15. This is the last row in our table. You see that the structure weight increases and the penalties decreased a bit. So we come much closer to the global optimized parameters, what we wanted to achieve. Therefore, you see we find a solution on how to use the global optimized parameters, including a position-based penalty, to achieve a good local alignment quality result. Of course, we also wanted to see whether the boundary detection and the structure weight trade-off would be improved by the position-wise penalty. For this, you can see here again the sensitivity and specificity, where the sensitivity again shows how well the non-coding RNA or your local motif is found, and the specificity shows how much of the context is not aligned. And we see this here for different penalties, so for penalty 0 until 30, and for all values are computed with a fixed structure weight of 200. And the non-coding RNA, so the local motif, is less and less covered once you increase the penalty, but also the context is not that much covered anymore by increasing the penalty. Comparing this result to what we have seen before for the structure weight, you can see the results now on the right side, shows you that we had the complete opposite situation. So for the sensitivity, the coverage of the local motif, for an increasing structure weight, you find more and more of the local motif, but you also tend to align into the context. This shows that the penalty can really compensate for the structure weight trade-off. Finally, of course, we also wanted to compare our suggested scoring to the default scoring. Our suggested scoring is now the global optimized parameters with structure weight 200. And the alignment quality here is calculated by the max SPS, which scores the correct alignment columns, but also penalizes whether the alignment was extended into the context. And if you compare the default scoring to the suggested scoring, you see that the penalty and the optimized parameters can improve the alignment quality prediction a lot. So today we have seen that there is a positive scoring bias due to structure. This bias leads to alignment length extension for random sequences. And we also have a trade-off for the structure between finding the complete non-coding RNA and aligning into the context. This trade-off can be compensated by setting a position-wise penalty, and this position-wise penalty and optimized parameters improve the overall alignment local quality. With this, I want to thank everyone who was involved in this study, Rolf and Milad from my group, Sebastian, who also really helped a lot and is the father of Locana, Frank Hutter and Ivo Hofacker, who also were very much involved in the thought process. Thanks for your attention, and now I'm open for your questions.